Okay, welcome. Hi, everyone. And thank you for being here. And thank you in particular to the organizer for giving me this opportunity. I'm Marta from the High Field NMR Center in Lyon. And I will present you today our magic angle spinning NMR investigation of the closed state of a bacterial cation channel. Now, from a, sorry, I have a problem with my cursor. Sorry, looks like I can't change the slides as I wish. Okay, let's try again. Okay, from a simplistic point of view, we often think about transmembrane channel as existing in two limiting states. As I call here, an on-off state model, where we have a closed state or off state and an open state or on state. And this model is actually applied also to describe the channel that I want to show you today, which is Corée, a pentameric channel, which is the major magnesium uptake systems in prokaryotes, a 200 kilodalton protein. For Corée, we have, from X-ray and cryo-EM, we have structures obtained in the two limiting conditions, that is at high concentration of magnesium or metal ion in general, and at low concentration of metal ion. And from these structures, a model, an on-off model, has indeed been obtained, in which we think of Corée as existing as a rigid, dry, and closed state at high concentration of metal ion, and a flexible, hydrated, open state at low concentration of metal ion, and also with a complete loss of symmetry upon magnesium removal. But now this picture is actually obtained, as I was saying, from X-ray and cryo-EM, which are often performed in detergent and at cryogenic temperatures. What we want to do is to investigate this system with magic angle spinning NMR, which is a very powerful technique for membrane proteins. Indeed, we can, after purification, we can reconstitute our membrane protein in lipid bilayer and directly centrifugate into the MAS rotor. And today, thanks to the current development in the NMR instrumentation, we don't need to rely on carbon detection, which is affected by low sensitivity, but we can instead use proton detection in fully protonated protein, which combines elevated high sensitivity and atomic resolution, using only some milligram amount of sample. So overall, with magic angle spinning NMR at fast mass, we can investigate our membrane proteins in a native-like environment with atomic resolution and with short experimental times. If we use this approach on the two limiting state of Corée, that is in the presence of high concentration of magnesium and in the absence of magnesium, we observe that the fingerprint spectra to decorrelation spectra are actually very similar. And this is quite puzzling, as with respect to the on-off model proposed by X-ray and cryo-EM. So we can wonder if actually this model is sufficient to understand the behavior of this channel. And this is what drove us to start investigating this system. And today, in this short talk, I will only focus on the first part of this work, which is related to the form obtained at a high concentration of metal ions, so the magnesium-bound form, that we investigated with ultra-fast magic angle spinning in lipid bilayer and at physiological temperatures. And I will show you that actually this state in this condition is not rigid and dry as predicted by the on-off model, but is instead dynamical and hydrated. The use of fast magic angle spinning and diamagnetic field leads to very high quality spectra for this very large protein. We have a pentamer of 360 residues, and due to the optimal spectral quality and long coherence lifetimes, we can go for 3D spectra for resonance assignment. In particular, due to the large size of the system, we here just show an example of strip plots, but actually we acquired a full data set of experiment relying on the detection of amide protons and H-alpha protons with a methodology which have been developed in my group over the years. And these actually allowed to have fewer assignment ambiguities. And we can also rely on a routine for automatic assignment, which is today adapted to the full solid state data set. And thus we can obtain our backbone resonance assignment. And in particular, we have extended assignment in two important regions of the protein, the transmembrane domain and the alpha-beta-alpha domain in the cytoplasm region. And in particular, the transmembrane domain is very important because it's the entrance gate for magnesium in the membrane. And we want to investigate the solvent exposure of this region. And to do that, we played a bit with the proton content of our samples. Here you can see in the spectra that a comparison of a 2D correlation spectra of fully protonated core A in water, here in blue, and the fully protonated core A that we washed in D2O. As a result, in this red spectrum, we can only see the peaks of residues which are non-exchangeable with solvent. And I labeled here the peaks which are located in the transmembrane domain, and as you can see, they are all present in this red spectrum. We can confirm this information with a deuterated sample that we then, in which we then introduce proton exchange. And in this case, as we don't use any refolding protocol, we only observe the exchangeable residues. Now in 2D, we don't have enough resolution, but if you go for 3Ds and observe the CN projections, it is clear that the transmembrane residues are actually missing in the deuterated, in the spectrum of the deuterated sample. This indicates that the transmembrane portion of core A does not exchange with water. But this doesn't mean that actually the transmembrane pore is dry. Indeed, we could, when comparing the relative intensity of the peaks of the sample of protonated core A in D2O and in H2O, we obtain an interesting correlation with the size of the pore shown here on the right. And in particular, we could observe that in the regions where the pore is narrower, we have the higher relative intensity, while in the regions where the pore is broader, we have the lowest relative intensity. And this suggests that actually a slow passage of water is allowed in the transmembrane pore, indicating that this is actually hydrated. Now, I was saying that we have some assignment, some extended assignment into defined region of the protein, but as you can see in this figure where I show the assignment on the structure of core A, we have missing assignment in the central part of the protein. And actually, and a large portion of peaks which are located in this region are missing in the spectra or not linkable. Nonetheless, it is possible to extend a bit the assignment and to predict some additional assignments for isolated residues using other information such as chemical shift index. And investigating site-specifically all the assigned residues, it is possible actually to understand the reason for the absence of a large number of peaks in the structure and understand the behavior of magnesium-bound core A. So our idea is that this effect is actually due to dynamics, as indeed a large number of motions can occur in the protein over a wide range of time scale, from local motions to large collective motions. This information is actually missing in the structural determination, but instead, magic angle spinning NMI is a powerful technique to access protein dynamics because it can rely on a variety of techniques which have been developing over the years. So this is a very active field. Today, in particular, I will show results in terms of nitrogen relaxation rate, so for backbone dynamics, and in particular, R1 draw, and nitrogen relaxation dispersion, which can overall cover a wide range of motional timescale. We access the site-specific determination of relaxation rates, introducing relaxation evolution blocks in our 2D correlation experiments, or since we have a very large system, we can also implement this on a 3D experiment to increase the resolution. Starting with nitrogen R1 draw, we could measure relaxation rates for a large number of residues in the protein. And what is intriguing here is that the residues which are located in the central parts of the protein are indeed the one with highest relaxation rates, indicating that the central region is likely the most dynamic region of the protein. And this information is actually also confirmed on a faster time scale from the measurement on nitrogen R1. Even more interestingly, it's the determination of relaxation dispersion, the acquisition of relaxation dispersion experiments, as these are very sensitive to the presence of conformational exchange with invisible NMR states. So we acquired relaxation dispersion experiments on deuterated core A, and we could observe that while for a number of residues here shown in blue, we have flat profile, indicating the absence of exchange, for a larger number of residues located both in structured and unstructured part of the protein, we have a relaxation dispersion profile, indicating the presence of such exchange. And this is particularly true, again, for these residues in the central part of the protein, which are both highly dynamic and involved in these exchange processes. So actually, surprisingly, magnesium-bound core A in magnesium-bound core A, the exchange between different conformation is accessible. So overall, with our investigation, we could move for an initial model in which we expect magnesium-bound core A to be very rigid and dry, to a more complex scenario where actually magnesium-bound core A is hydrated and can populate a more complex conformational landscape. And this is, as I say, is our starting point, but it's actually, this new scenario is can better elucidate also the gating mechanism as and provide a new view to understand the way magnesium is gated through the channel. So with this, I want to thank my group, in particular Guido Pintacuda, my supervisor, Tobias Schubeiser and Andrea Bertarello, who are involved in the project, all the people from the CRMN, and you all for your attention.
That was great. Thank you very much, Marta. So we're going to go straight on to Thomas now.
Hi, everybody. I'm excited to present here. I hope you can all see my slides. So I'm happy to present here. I am excited to present our new method for 2D distance reconstruction and post-dipolar EPR spectroscopy. And I am from the Marius Kroll Lab. This project was a collaboration with Jack Fried and especially Madhur Srivastava. And the method what we're going to present is not actually just only for EPR purpose. It actually can be applied to any form of spectroscopy. So I urge you after the talk to Madhur Srivastava. He is really the brainchild behind coming up with a method. And here we again. Before continuing, I want to just kind of introduce EPR spectroscopy for biological molecules, specifically PUDs, EPR spectroscopy for the audience, and how we actually go about it. So in our interest is normally a distance measurement. I'm going to introduce a method later on, but what we rely on is that there's a paramagnetic species. In many cases, we need to introduce this paramagnetic species, and we do that via a specific engineered cysteine, where we now attach a covalently linked paramagnetic label, such as a nitroxide. As a final result, we're going to get hopefully a large library of labeling sites. And just to visualize how that's going to look like, here we have a protein, a rather rigid protein for that sake, labeled with our paramagnetic labels. And you can see how the rotamers distribute the label in correlation to our large brother thread. So once we have the labels now attached and we do have our library, we can pick two of them, most likely only two, paramagnetic labels, and we want to determine the distance between them to identify, for example, domain movement or such alike. And for that reason, as I said, we attach two paramagnetic labels, and we now pulse on one of them, which is going to be our observer spin. We flip our pump on the other frequency, the pump spin, and based on their dipolar coupling between them, we can determine our distance distribution. So how does that look like? Well, we're going to obtain our dipolar evolution trace shown in red. We can do a background subtraction or such alike. And we will obtain our final signal S of t, which we now can use to identify our distance distribution P of R. Now that sounds very simple, right? It's rather straightforward. Here's a protein, label it, get a distance distribution, great. Now there's a tricky thing though. The tricky thing is really converting the S of t to a P of R, because it's an ill-posed problem. and we need to play some tricks on that end. So in our lab, at least, we're using majorly 3 different methods, and there are many other flavors, to be fair. And for that, again, we start with our dipolar signal S of t. We can apply any kind of different filters, in this case, maybe a wavelet, denoising, or any other filter for that sake. And now we can diverge. We can do a model fitting where we have a model, what we're interested in, we can fit it directly to our dipolar signal. Now this is great. However, that requires an a priori knowledge of what the model is, which is not always the case. So then we go over to the next methods, which is either de-analysis or de-lab nowadays, which is a model-free method and uses a global optimization. That means we optimize against the entire trace, or we're using the Srivastava-Fried SVD method, which again is a model-free method, but utilizes a local optimization for each individual time point. And the difference between them come really in that we're looking at either a Tikhonov regularization versus a Picard cutoff. So just to iterate that problem, we have a problem here. We have a signal, we want to get the population of the distances. Now, how do we go about it? We rearrange it, and now we're looking for a population of the distance. In the Tikhonov regularization, we depend on a regularization factor lambda, whereby in the SVD form of it, we handpick our sigmas based on our Picard cutoff. Now this is nice. Now you guys know all the magic behind it. I have a protein, put label on it, get my DO trace, and I can now convert the dipolar evolution line to the dipole, and now I get my population P of R, thanks to those grade 3 methods engineered. However, what are we going to do if we have, let's say, other variables? Let's say we have a protein kinetics issue. We are looking at multiple traces as a function of concentration or freezing time, where we have each frozen time point in the reaction coordinate. Or we have a protein unfolding. That means we have for each urea concentration a dipolar evolution time trace. Or for distance assignment, which I'll explain later on. Now, in this case, actually, we're introducing another dimension. And the other dimension is specifically, we can call it the urea for urea concentration dimension, or in case of protein kinetic, it's another time dimension in the reaction coordinates. So this one, we just designate overall as our trace dimension. If that happens, we can now go in 2 different ways. We can have our stack of dipolar signals shown in the middle right here. And we just label them S. And now from S, we can go 2 ways. The most common form, and I think this is a great form, it works wonderful, go from the each single trace, analyze them in parallel. That means each individual time trace is analyzed separately. Now, if we do that, we get each one where we get a distance population P of R, or P of Q. And however, each one will have a some differences due to the noise level variation in between the traces and differences in the different parameters. However, what we decided to do, and this was largely done like by Maduro Srivastava, who came up with that method, is to rather use, instead of a 1D SVD, we use a 2D SVD method. Now, in this case, actually, we stack all the traces on top of each other. We know there are some populations in there, and we think that, okay, all of those different time points are correlated in time with each other. So they are all seeing each other. So we get a nice little grid where we have, in one dimension, we have the time dimension, and in the other one, we have the trace dimension. And what we gain out of it will be a beautiful P of R, but now it's not as an individual trace. They're all correlated as a stacked, like a surface. We're actually getting a distance distribution surface out of that. And this is kind of neat. So how do we do that? It's rather straightforward in this matter. We have our signal, again, which is a time dimension, which we just designate M, and then we have our trace dimension, Q, and we formulate the problem to get our population as a function now of trace and time dimension, and we now can get our very stable answer for our sigma values, which we now then identify due to the Picard cutoff in a stable region or in the unstable region. And voila, we end up with a rather smoothened surface, distance distribution surface. However, the crux of the matter is that we actually should do it with an experiment. I think this is much better. The truth of the matter is in the pudding. So we went ahead and we used our T2 edited deer experiment, or TM edited. And now I don't want to delve too deep what the experiment really is. However, I'm just going to briefly introduce it for the community. In this experiment, really what it is, a T2 edited, is actually really similar to a PRE experiment by NMR. So in NMR, what do we do in a PRE experiment? We're looking at nuclei, or proton for that sake, and upon introducing a paramagnetic species, the relaxation of those nuclei is enhanced. Now here we just do the reverse. We're looking now at an electron, and upon close proximity to a protonated species, its relaxation is enhanced. And that really manifests itself if we look at the distance distribution. So here we have a bimodal distribution, one of them deriving from one distance, the other one from the other one. Now, this specific distance distribution comes in close proximity to those losing protons. And so we can see a rapid decrease in its population. So again, this is really close to a PRE experiment, almost identical. And for that sake of argument, I'm just going to label PRE. Instead of paramagnetic relaxation enhancement, I'm going to say proton relaxation enhancement. And we use this experiment to just validate the 2D SF-SVD method. So therefore, we used our model protein A. We deuterated it, and we attached protonated leucine residues into the deuterated protein A. We have our paramagnetic labels shown in red and orange. We measure our distance once from A red to orange, or from B red to orange, and we will get the dipolar distance distribution like shown here. Now, if we go ahead with our PRE measurement, we will see that one of the distance remains rather stable, and the other distance is decreasing in population as the time goes on. Again, identical to a PRE measurement in NMR. And we analyze now the time traces with one time the 1D SF-SVD method and the other one the 2D SF-SVD method. In such a matter, you will see right away that the one decrease has its tails, has valleys, so it's very ragged. However, upon applying the 2D SF-SVD, it smoothens out. So if I would be a skier and I would be in the Alps or the Rockies, I'd probably pick the more nicer slope. I don't want to go in the valleys. So the same occurred for another model system. Again, same protein, same labels, but now in this case, we actually introduced protonated phenylalanines into our protein, and we see a similar effect that we're going to see a decrease. However, it was not as obvious, and the data was a little bit more noisier due to its nature. And what we can see again, we see is similar to the previous example. Once we apply the 1D SVD, it's very ragged. But once applied the 2D SVD, it smoothens out. It smooths out rather dramatically, to be honest. And how to quantify, quantify the matter, we just took the overall peak height and made the ratios of both of the peaks. So meaning the one which is stable compared to the one which is decaying. And you can see that upon applying the 1D SVD, you can see it's more ragged. However, upon applying the 2D SF-SVD, it really smoothens out a lot for the leucine sample, and it really helps to like the phenylalanine sample as well, to see what's going on with a decay. So this is a very beautiful example where the 2D SF-SVD did its job, and it truly increased the resolution. Now, as a conclusion, what I want to drive home is 2D SF-SVD permits reliable reconstruction of 2D distance distribution for pseudo 2D DO traces. So that means if you're interested in protein kinetics and protein unfolding, you want to look at EPR by DO traces, or you have a fancy pull sequence which has introduced another dimension, give this one a shot. It could help you. If you're in NMR and you have some pseudo 2D data or pseudo 3D data, look into the method. It could help you out a lot. It might solve your problem. And just to iterate again on what's going on, here we have just some simulated reaction coordinates where I go from species A to B and finally to C. I simulated, thanks to DeoLab, I simulated those deotraces at various noise level. And you can see, you can still, even at very high noise level, you can still get a good amount of information out of the traces, thanks to the 2D method. And at last, I want to say thank you. Thank you for listening. Thank you to you. And especially thank you to Madhur Srivastava that he came up with that. And Jack Fried for helping out. And then obviously to the big cheese himself, Marius Chlor, and the entire Chlor group, and various other people. And at last, I want to say thank you to the International EPR Society, which is really the glue behind the EPR community. And thank you very much.
Well, thank you very much, Thomas. So we're going to go straight on to our final speaker before breakout rooms. So Xiuxiu, when you're ready, thank you.
So you can see my screen, right?
Yep,
hello.
That's good.
Okay. Hello, everyone. Thanks, organizers, for giving me this opportunity to talk about my work. Today, I'm going to talk about proteasome subunit RPN13 interaction with ubiquitinated substrates and proteasome. RPN13 is a ubiquitin receptor. It is bound to the proteasome through RPN2. It's composed of a pro and a dual domain. Pro used to bind ubiquitin chains, and proteasome. Dual domain is used to recruit ubiquitinase UCH37 and activate its hydrolase activity. When I started this project, only the crystal structure of its RPN2 was reported, but the binding region of RPN2 to RPN13 is unknown. I was able to use ARMA and chemical and biophysical assay to figure out RPN2 extremely C-terminal 940 to 953 is used to bind RPN13 with 27 nanomolar. This strong binding affinity enables more than 200 intermolecular OE detected. So I calculated this complex structure by using read trend generated from intra and intermolecular OE. RPN13 is colored in purple and RPN2 is colored in light orange. So as you see here, RPN2 stretches across a narrow canyon of RPN13 pro domain. Ubiquitin binding region is colored in yellow. So we can see RPN2 and ubiquitin bind to different locations of RPN13. RPN2 zippers along RPN13 surface through a proline-rich region. This data line represents experimental NOE data involves RPN2 proline region. So this is selected experimental data acquired on carbon-13 labeled RPN2 with equal molar of unlabeled RPN13 pro domain. It's focused on RPN2, these three prolines with intermolecular OE to RPN13 tryptophan 108 aromatic protons. RPN2 to aromatic residues form hydrophobic interactions with RPN13 pro domain. So this data line represents selected experimental NOE data for these two aromatic residues. This is selected NOE data acquired on carbon-13 labeled pro with equal molar of unlabeled RPN2 peptide. At that time, and until now, 4 years later, couple of resolution human cryo-EM structure have been reported, but no any information for human RPN13. But I was able to document complex structure to its cryo-EM density. So you see here, the fitting looks good. But how does RPN13 bind to K48 linked ubiquitin chain has not been resolved. So I made a series samples. Before that, I want to clarify what's proximal ubiquitin and distal ubiquitin. So this is K48 diubiquitin. Ubiquitin has a C-terminal free can be attached to substrates called proximal ubiquitin. Ubiquitin C-terminal form isopeptide with lysine 48 of a proximal ubiquitin called distal ubiquitin. So the first sample is RPN13, is carbon-13 labeled with equal molar of unlabeled RPN2 peptide with 1.2-fold of K48 diubiquitin. Distal ubiquitin is carbon-13 labeled. So proximal ubiquitin is colored in yellow and distal ubiquitin is colored green. So I will keep this coloring throughout the whole presentation. So I ran carbon-13 half-life noding on this sample. So this is carbon-13 labeled RPN13 residues. So we can see a lot of intermolecular OEs formed between pro and proximal ubiquity. To make it simple, I just use RP13 isoleucine 74 as an example. So isoleucine 74 methyl groups form intermolecular OE to proton attached to carbon 12 of a proximal ubiquity, residues isoleucine 44, valine 17, and also glutamine, beta and gamma protons. I also ran carbon hyphenol on this sample, but with proximal ubiquity carbon 13 labeled. A similar intermolecular OE was observed between RP13 pro and the distal ubiquity. By using the intermolecular OE between RP13 pro and the peptide as a control, we can see RP2 RP13 has a strong intermolecular OE to proximal ubiquity. No matter what, RP2 RP13 can bind to each ubiquity, but with preference for proximal ubiquity. Finally, I got 72 or 56 intermolecular OE between RP13 pro with proximal ubiquity or distal ubiquity. So I calculated these two structures. The bound ubiquitins are convergent in the 15 lowest annual structures. I superposed these two structures. The superposition of bound ubiquity, RP13 pro, RP2 peptide are good with RMSD 0.8 angstrom. Due to similar intermolecular OE, RP2 bound RP13 binds to proximal ubiquity or distal ubiquity with common mechanisms. So here I'm just show you the interactions between RP13 pro and the proximal ubiquity. RP13 leucine 56, phenylalanine 76, and the isoleucine 74 form hydrophobic interactions with proximal ubiquity leucine 8, valine 17, and isoleucine 44. So these three residues are for ubiquitin, also used to bind other ubiquitin partners. RP13 also form extensive interactions with K48 diubiquitin linked region, including alanine 46, glycine 47, and lysine 48. A hydrogen bond was formed between RP13 lysine 103 subchain and distal ubiquitin glycine 76 backbone. Surprisingly, the interactions between proximal ubiquitin and distal ubiquitin were also observed. So this is the selected MR data of carbon-13 hyphenol acquired on this sample. So this is the carbon-13 labeled proximal ubiquitin residues. You can see a lot of NOEs formed to distal ubiquitin leucine 73. So in the structure, you can see leucine 73 form extensive interactions with proximal ubiquity centered on tyrosine 59. So these inter-ubiquitin interactions caused a little bit of restrictions to the unbound ubiquitin. So as you can see here, unbound ubiquitin is not going everywhere. It's in the limited orientation due to this a little bit of restriction caused by distal ubiquitin leucine 73. So we were wondering whether this new conformation is induced by RP2 bind RP13 or is already existing in the free solution of K48 diubiquitin. So I ran carbon-13 hyphenol on free K48 diubiquitin with proximal ubiquitin carbon-13 labeled. So this is the carbon-13 labeled proximal ubiquitin residues. So you can see formal NOE to distal ubiquitin residues. So this strong NOE dataset supports the reported closed conformation of a K48 diubiquitin solved by X-ray. In this structure, proximal ubiquitin is colored brown, distal ubiquitin is colored in blue. So for example, distal ubiquitin glutamine 49, proximal ubiquitin and glutamine 49 form NOE to distal ubiquitin arginine 72 protons. In addition to this strong NOE dataset, a small population of NOE also were observed. So this NOE dataset matched the NOE observed in the presence of RP2 bind RP13. And also you can see this NOE dataset actually was enriched in the presence of RP2 bind RP13. But the NOE dataset supporting the closed conformation disappeared. It indicates in the free solution of K48 diubiquitin In addition to this dominant closed conformation of K40 diubiquitin, also this small population of new conformation discovered in the presence of ARPin2 bound ARPin13. With ARPin2 bound ARPin13, it will shift the equilibrium by binding to proximal ubiquitin or distal ubiquitin, but with preference for proximal ubiquitin. Here we can see ARPin2 bound ARPin13 can exchange dynamically with each ubiquitin. So this also happens to other ubiquitin receptors. So maybe the dynamic nature of ubiquitin receptors to ubiquitinated substrates may add in orienting of ubiquitinated substrates for downstream processing, including deubiquitination by deubiquitinase and offloading by ATPase. I'm grateful to my supervisor Kelly and my lab members and our collaborator Hiroshi, and thanks our founding intramural research program. Thanks Kevin Gardiner linked me to this bigger NMR facility. Thanks everyone. I'm happy to chat with you in breakout session. Thank you.